Wow, 1 in 36.
It's a striking number, isn't it?
Yeah, 1 in 36 children in the U.S. identified with ASD.
Autism spectrum disorder, yeah.
It really makes you think about all the different factors at play. And, for so long, the focus has been on neurons, the brain cells responsible for communication.
Right, the wiring of the brain.
But what if there's another piece of the puzzle, something we haven't fully considered?
Well, there's growing recognition that the brain is a much more complex ecosystem than we initially thought.
Okay, so today we're diving into some groundbreaking research that sheds light on a potential new player in ASD. And we're going to be looking at the brain's immune cells,
Microglia.
microglia,
Yeah.
right? And how they interact with neurons, especially when a specific gene linked to ASD isn't working as it should.
This research really highlights the intricate relationship between the nervous and immune systems in the brain. These neuroimmune interactions, as they're called, are crucial for healthy brain development.
So let's lay out what we'll be exploring today. First, we'll break down this gene, SCN2A, and why a problem with it is so strongly associated with ASD.
It's one of the leading genetic causes, actually.
Then we'll look at how the researchers studied this using both mouse models and these amazing lab-grown structures that mimic the human brain.
Cerebral organoids, they're incredible tools for research.
And then we'll get into the surprising role that microglia seem to play when this SCN2A gene isn't functioning correctly.
It seems they might be a bit overzealous in their duties.
We'll talk about how these immune cells become overactive and what that means. And finally, we'll discuss the potential implications of these findings for understanding and maybe even treating ASD.
What's particularly fascinating is that the SCN2A gene is mainly active in neurons. This allows us to see how a change in those nerve cells can trigger responses in other cell types, like the microglia.
This is what really intrigues me, because it's not just about confirming what we know about how the brain is wired, but it suggests that the brain's cleanup crew might be reacting in an unexpected way when that wiring isn't quite right.
A very apt analogy.
So are you ready to dive into the details?
Absolutely. Let's start with the SCN2A gene itself.
SCN2A, it sounds very scientific. What exactly does it do in the brain, and why is it so significant when we talk about ASD?
Okay, so SCN2A is basically the instruction manual for building a specific type of channel on the surface of neurons called a sodium channel.
A sodium channel.
Yes, specifically the NaV1.2 sodium channel. These channels act like tiny gates, controlling the flow of sodium ions in and out of the neuron.
I see.
And this flow is absolutely crucial for neurons to generate electrical signals, which is how they communicate with each other.
So they're like the switches that allow information to flow through the brain circuits.
Exactly. If the SCN2A gene isn't working properly, if it's not producing enough of these functional sodium channels, then the communication signals between neurons get disrupted.
And that's where the link to ASD comes in, right?
Precisely. As this study highlights, SCN2A deficiency is considered one of the leading single-gene causes of both ASD and intellectual disability.
So a problem with just this one gene can have a huge impact.
It can. In their research, they used a mouse model that lacked the mouse version of the gene, which is called SCN2A, and they observed significant difficulties with learning and memory in these mice.
How did they actually measure learning and memory in mice?
Well, they used some standard behavioral tests like the Morris water maze.
I heard of that one.
Yeah, the mice have to learn the location of a hidden platform in a pool of water. And the mice with the SCN2A deficiency took much longer to find the platform, and they didn't improve as much over time compared to the control mice.
So they had trouble remembering where the platform was.
Right, which indicated a problem with forming long-term memories, specifically about spatial locations.
Fascinating.
They also used a Y maze, which tests short-term spatial working memory. That's the ability to remember recently visited locations.
And did the SCN2A deficient mice struggle with that too?
They did. These tests clearly showed a tangible impact of the SCN2A deficiency. The mice struggled to learn and remember things.
So they had these cognitive issues because of the SCN2A problem, but the study went deeper than that, right? They actually looked at the connections between the neurons themselves.
Exactly. Learning and memory impairments are often linked to changes in synaptic transmission, which is how neurons communicate at their connection points, the synapses.
Right, the synapses.
They used a technique called electrophysiological recording to measure the electrical activity at these synapses in the hippocampus, a brain region crucial for learning and memory.
Right.
And they found that the strength of these communication signals, specifically what are known as miniature excitatory postsynaptic currents, or MEPSCs for short, was significantly reduced in the SCN2A deficient mice.
MEPSCs, got it.
But interestingly, this reduction was only seen in later stages of development and adulthood, not in very young mice.
So the problem emerged over time.
It seems so, suggesting that the initial wiring of the brain might be relatively normal, but something goes wrong during the ongoing development or maintenance of these connections.
That's a key insight.
To investigate this further, they looked at the physical structures of the synapses by counting the points where the sending and receiving parts of the synapse overlapped.
So they were actually looking at the physical connections.
Yes, and consistent with their electrical measurements, they found fewer of these connections in the hippocampus of the SCN2A-deficient mice in the later developmental stages.
Interesting.
They also observed a lower density of dendritic spines on the neurons, which are the little protrusions on the receiving end of the neuron that form the connections with other neurons.
So fewer connections overall, and this is where the research took a really clever turn, right? They started growing these neurons in a dish all by themselves without any other brain cells around.
Precisely, by culturing neurons from the SCN2A-deficient mice in vitro, they could isolate the direct effects of the gene deficiency on the neurons themselves.
Makes sense.
And while they did observe that the dendritic spines on these isolated neurons appeared less mature, they didn't find a significant difference in the overall number of spines compared to the control neurons grown in isolation.
So the neurons themselves, when isolated from their usual brain environment, don't fully account for the reduced number of synapses seen in the whole animal.
That's right, which strongly suggested that other cell types in the brain must be involved.
Like the microglia.
Exactly. This difference between what they saw in the living brain and what they saw with isolated neurons led them to suspect that microglia might be playing a significant role in the reduced synapse density in the SCN2A deficient mice.
So for those of us who aren't brain experts, who exactly are these microglia? What do they normally do in the brain?
Microglia are the specialized immune cells that reside in the brain. They act like the brain's internal surveillance and maintenance team.
Okay.
One of their crucial roles is to help regulate brain development and keep it healthy by doing things like clearing away cellular debris and, very importantly for our discussion, pruning synapses.
Pruning synapses.
Yeah, synaptic pruning is a natural and necessary developmental process where the brain eliminates excess or weaker connections between neurons to refine the neural circuits and make them more efficient.
It's like the brain is trimming away the extra branches to help the signals flow more smoothly.
Exactly. But in the SCN2A deficient mice, the question was, could the microglia be going a bit overboard with the pruning?
Like overdoing it.
Right, so they examined the shape and the activity of microglia in the hippocampus of these mice, and they found that the microglia had an altered shape with larger cell bodies and fewer of those fine branch-like extensions they usually have.
That sounds significant.
This change in shape is often a sign that the microglia have become activated. Interestingly, they didn't see a major increase in the number of microglia, but they did observe a significant increase in the size of lysosomes within the microglia.
Lysosome.
Lysosomes are like the recycling and waste disposal centers within a cell.
So if the lysosomes are larger, that means the microglia are taking in and breaking down more material.
Exactly. This pointed towards increased phagocytic activity, the process by which cells engulf and digest other cells or cellular components.
And in this case, they suspected it could be synapses.
Right, and they actually found direct evidence for that. Using a technique called immunofluorescence microscopy, they looked for the presence of synapses inside the microglia's lysosomes.
Very clever.
They discovered a significant increase in the engulfment of postsynapses by microglia in the hippocampus of the SCN2A deficient mice.
Postsynapses, that's the receiving end of the neuronal connection.
That's right, and interestingly, they didn't see a similar increase in the engulfment of the presynaptic side, the sending part of the connection.
So it seems like the SCN2A issue in the neurons is somehow signaling the microglia to come and excessively prune those postsynaptic connections. But what could be acting as that signal?
Well, they investigated the involvement of the complement cascade, which is a part of the immune system that's known to tag synapses for pruning by microglia during normal brain development. Specifically, they looked at two complement proteins called C3 and C1q, and they found increased levels of both C3 and C1q, specifically located on the postsynapses in the hippocampus of the SCN2A deficient mice, precisely at the developmental stage when they observed the excessive pruning.
So it's like the neurons with the SCN2A problem are getting flagged with these complement proteins, and that's acting like a signal for the microglia to come and remove them, like an eat me sign.
Exactly. To further test this idea that microglia were directly causing the synapse loss, they used a drug called PLX3397 to selectively eliminate microglia in the SCN2A-deficient mice.
So they got rid of the microglia. What happened then?
The results were quite remarkable. They found that depleting the microglia partially restored the normal level of synaptic transmission as measured by the frequency of those MEPSCs we talked about earlier.
Right.
It also partially restored the density of dendritic spines in the hippocampus of the SCN2A-deficient mice, which strongly suggested that the overactive microglia were indeed responsible for at least a portion of the synaptic problems.
That's a really powerful finding, but they didn't stop there.
Yep.
We wanted to see if this was also relevant to understanding autism in humans.
Of course, to make that crucial link to humans, they used human-induced pluripotent stem cells, or HIPSCs.
HIPSCs.
These are cells that can be reprogrammed to become any cell type in the body, including brain cells.
Wow.
They use these to create cerebral organoids, those three-dimensional miniature brain-like structures grown in the lab that can mimic aspects of human brain development.
It's incredible what science can do these days.
It is. They engineered these organoids to carry a specific SCN2A mutation that's been identified in children with autism, and then they introduced human microglia, derived from healthy iPSCs, into these organoids.
So healthy human immune cells interacting with human brain tissue that has the SCN2A genetic issue.
Exactly, and what they observed was fascinating. The human microglia within the organoids carrying the SCN2A mutation showed signs of activation, including increased size and increased presence of a marker called CD68, which indicates increased lysosomal content.
So similar to what they saw in the mice.
Precisely, and crucially, mirroring their findings in the mice, they also observed an elevated engulfment of postsynaptic markers by these human microglia in the SCN2A mutant organoids.
So this excessive pruning by microglia in response to SCN2A deficiency isn't just limited to mice, but it's also relevant in human brain tissue.
That's a key takeaway. This study provides compelling evidence for a significant role of microglia in the biological processes associated with SCN2A deficiency, a major genetic contributor to ASD.
So it really broadens our understanding beyond just neuron-specific issues.
Right. It highlights the critical importance of these neuroimmune interactions, even when the primary genetic defect is in the neurons.
So stepping back for a moment, what does all this mean for how we understand ASD?
Well, in the wider context, this research suggests that in some forms of autism, even when the primary genetic issue originates in neurons, the brain's immune cells can play a substantial role in shaping how neural circuits develop and function.
It's a whole new perspective.
It opens up exciting new possibilities for developing therapeutic interventions that might target microglial activity to help restore a more balanced level of synaptic pruning.
It's amazing to think about the potential for future treatments.
Absolutely. This research adds a really important layer to our understanding of a complex condition. It underscores the fact that autism is a very complex condition with diverse underlying mechanisms.
Right, it's not just one thing.
And gaining a deeper understanding of these different pathways will be crucial for developing more effective and targeted approaches in the future.
This study provides a significant piece of that puzzle. Could understanding and modulating microglial activity represent a new frontier in addressing the underlying mechanisms of some forms of autism?
It's certainly a question worth exploring further. And what are the ethical considerations of manipulating the brain's immune system?
That's a great point. There's always more to consider. Well, this has been a truly fascinating deep dive into this groundbreaking research. Thank you so much for breaking down this complex science in such an understandable way.
It's been a pleasure.
And thank you all for joining us as we explored this intriguing connection between a key autism-linked gene and the brain's immune system. We hope you'll continue to explore the ever-evolving world of brain research with us.
Until next time.
See you then.